So in the first part of our search for genetic material, we ended in 1944 with Avery, MacLeod, and McCarty's confirmation that bacteria is transferring DNA during bacterial transformation. And these findings made DNA a strong candidate for genetic material, but they were not enough to convince scientists because at that time, most scientists believed that protein was a much stronger candidate for genetic material. So along came Alfred Hershey and Martha Chase in 1952, who settled the question of protein or DNA once and for all. So they knew that bacteriophages, viruses that infect bacteria, somehow could use their genetic material to reprogram the bacteria they infected. So was it the bacteriophage's protein or was it their DNA that was responsible for reprogramming the bacteria? Hershey and Chase grew two different bacteriophage cultures, one in radioactive sulfur and one in radioactive nitrogen. And the logic behind this, sulfur is only found in protein, whereas nitrogen is mainly only found in DNA. And so by growing these bacteriophages within these radioactive solutions, one would incorporate the radioactive sulfur into its protein and end up having radioactive protein, whereas the other would incorporate radioactive nitrogen into its DNA and end up having radioactive DNA. So, after the radioactive bacteriophages infected the bacteria cells, the cells were agitated in a blender. And what this did, this is a blender, it separated the bacteriophages from the cells. And so here you see in this tube, the bacteriophages were in the top half, in this section of the tube, and the cells were sort of collected into the bottom half of the tube in a sort of pellet. And what they found, the bacteriophages that had radioactive protein, the radioactivity all stayed at the top of the tube. So it was not within the cells, whereas the radioactive DNA bacteriophages, all the radioactivity ended up in the bottom of the tube with the pellet, with the cells. And so what that means, the radioactivity of the DNA or the DNA itself had somehow transferred into the cell. And this confirmed that the DNA had transferred from virus to cell, or in other words, that DNA, not protein, is the genetic material of bacteriophages and viruses. And these results later prompted further experiments that confirmed nucleic acids, DNA, and RNA are the hereditary material for all organisms. And this is where we end our second video. Please be sure to check out our third.